In the early 30s, a husband and wife team coined the phrase essential fatty acids. Until then, dietary fat was viewed simply as a source of calories. A decade later, a British physiologist, Hugh Sinclair, made his first visit to the Inuit. He hypothesised, in a famous letter to The Lancet in 1956, that their diet, rich in omega-3 fatty acids, was protective against Western diseases such as atherosclerosis. In the 70s, he found, with the Danish investigators Bang and Dyerberg, that the incidence of myocardial infarction was 10 times lower among the Inuit compared with the Danes. Those observations were the backbone for the development of the first pharmaceutical omega-3. Omacor is the first omega-3 acid ethyl esters derived prescription drug approved by authorities as a supplement to the diet for the treatment of specific hypertriglyceridemia subtypes and in secondary prevention after myocardial infarction, in addition to other standard therapy. We went to Kalundborg, Denmark, one of Omacor's production plants, to understand how fish oil becomes a pure and potent pharmaceutical drug. Tankers deliver the crude fish oil, which is directly pumped into big tanks. The small pipe on the side contains nitrogen, an inert gas used to prevent oxidation. Its key safety point is to avoid oxidation of the oil. At this stage, the crude oil contains about 30% omega-3 fatty acids, triglycerides, and a mixture of cholesterol, other fatty acids, and impurities. At the end of the manufacturing process, concentration will rise to more than 90% and oil will be purified from virtually all pollutants. Oil is fractionated and refined through an 8-step purification and concentration process, which takes approximately 3 weeks. The first step is deacidification, where free fatty acids and water-soluble impurities are removed. The free fatty acids reacts with sodium hydroxide. The reaction converts the free fatty acids to soap. The soap is separated by centrifugation and removed from crude oil. Already, at this stage, we can see that the colour of the oil becomes clearer. The oil is now ready for the second step in the process. The purpose of stripping is to reduce the contents of fat-soluble environmental pollutants like PCBs and dioxin, as well as vitamin A and D and cholesterol. In the third step, transesterification, the stripped oil reacts with ethanol to convert triglycerides into ethyl esters. This makes it possible to increase the concentration of EPA and DHA by distillation. Ethyl esters with molecular weights lower than EPA and DHA are evaporated, condensed, and removed as a light fraction, reducing the amount of short-chain fatty acids and increasing the concentration of EPA and DHA. Urea complexation removes saturated fat and non-omega-3 unsaturated fatty acids. The concentration is increased to about 70% of omega-3 acid ethyl esters. Then bleaching removes oxidation products from the oil. A second distillation is performed to achieve the final concentration of 90% omega-3 acid ethyl esters. The last step consists of mixing and drumming, where an antioxidant is added to the batch. From this tank, oil is ready to be filled into drums. We are now in the heart of the plant, the control room. Here, employees are able to monitor all the manufacturing steps. We asked the site manager to talk about the PCS, the process control system.
We have a very sophisticated PCS system, and we actually won the Amazon Prize in 2009 for the best utilization of our system, which is called Delta V. So it is really state-of-the-art how we adjust the processes and how we control the processes.
At different points, samples are taken and tested to ensure the right quality of the intermediate and final product. The central lab has different purposes in order to control the quality and purity of the product. These are a high concentration of EPA and DHA, a very low content of oxidants, a low content of environmental pollutants. The drums in the warehouse are ready to be sent to our encapsulators. We are now in Ploemel in France to examine the encapsulation process. Capsules are manufactured according to precise specifications which ensure the purity and quality of the product. In this room, drums are identified and connected to a vacuum hose. This tank corresponds to the production of about 570,000 capsules. The gelatin used to make the capsules is heated to 65 degrees Celsius and presented as a ribbon. The product is injected through pipes and the capsules are moulded. The capsules move to the drying rack and pass from one drum to another for about 2 hours. Tracking software traces the status of the product at each stage of the process. An operator regularly measures the total weight of the capsule and its envelope. The entire process was physically inspected by the FDA during its commissioning and is audited regularly by local and international authorities. Finally, capsules are packed in batches and sent back to Denmark for packaging. 2 million capsules arrive twice a week from the manufacturer. Each batch has a unique number, allowing tracking and quality control. Bottles are transferred to a counting machine. Capsules are counted with a photocell and a precise amount is delivered into each bottle. Labels and legal information are added. Every 10 minutes, an operator performs a quality control check. Boxes are delivered to the warehouse. The product is now ready for global distribution.